Antibiotics. Let us just have a fast review about the bacterial cell wall. Bacteria is a single cell microorganism that classified into two, depending on their response to the gram stain test. In here, we have the so-called gram-positive bacteria, meaning they are positive for gram stain test, and we have here the gram-negative ones that are obviously negative for the gram stain test. As you could see, structurally, they have different composition in their cell walls or their cell membranes. They have different layers to it. In gram negative, the peptidoglycan, which is the cell wall or the makeup of the cell wall of a bacteria, is way thinner compared to the gram-positive one. Antibiotics are drugs that kill or stop the growth of bacteria without harming the cells of the infected organism. This is derived from living organisms, although they are often made more often effective by various chemical processes. Nowadays, it is possible to get an antibiotic which is done synthetically in the laboratory. There are 5 ways on how antibiotics work. First one, they prevent the synthesis of bacterial cell wall. 2, they prevent the activity of proteins in the cell surface membrane of the bacteria. 3, they stop enzyme action of the bacteria. Number 4, they prevent DNA synthesis. And lastly, they also prevent protein synthesis in bacteria. One of the most used and most misused antibiotic is penicillin. Penicillin is...
You've probably cut your finger at some point in your life. Painful, sure, but no big deal. Well, imagine if that minor cut became so infected with bacteria that it killed you. Before the 1940s, that really did happen, a lot. The most minor injuries could be deadly. And then a discovery was made that changed the world. Penicillin. It was discovered totally by accident. In 1928, Scottish scientist Alexander Fleming left his lab to go on vacation without cleaning up. When he came back, he found mold had developed on a contaminated sample of staphylococci. Remarkably, the mold had killed the surrounding bacteria. He decided to name the substance released from the mold penicillin. But what is penicillin exactly? It is an antibiotic that fights bacteria by stopping the cell wall from renewing during growth. This weakens the cell wall until it bursts, killing the bacteria. It wasn't until the 1940s that America and Britain funded the mass production of penicillin. Hailed as a miracle drug that saved countless lives during World War II, it allowed the Allies to patch up a soldier and have him ready to fight again in a matter of weeks. By the end of World War II, US companies were making 650 billion units a month. Its discovery sparked the hunt for other antibiotics that are used today in everything from vaccines to cancer drugs. While the widespread use of penicillin has been a huge benefit to people across the globe, it might not be effective forever. Some bacteria have become resistant to commonly used antibiotics, and the overuse and misuse of antibiotics is leading to more bacteria developing the ability to defeat the drugs designed to kill them. In order to defeat these superbugs, scientists around the world are hoping to make a medical breakthrough, just like Alexander Fleming did 90 years ago.
So all in all, penicillin prevents synthesis of crosslinks between peptidoglycan polymers in the cell walls of bacteria. They cannot attack viruses since viruses do not have cell wall. Antibiotic resistance. Antibiotic resistance is good for the bacteria, but antibiotic resistance brings dangers to us human beings. Antibiotic resistance can occur because the bacterial protein can inactivate probably the effect of the antibiotic. 2, the bacteria can pump out the antibiotic that goes in their system, in the cytoplasm. And lastly, some antibiotics cannot bind to the target site within the bacteria. An example of this one is TB. We do not use penicillin in TB because the mycobacterium has an enzyme that catalyzes the breakdown of penicillin. If a bacteria is sensitive to the effect of the antibiotic, we call this one as susceptible to antibiotic. Bacteria has a genetic advantage over these antibiotics. It is because bacteria have only one copy of their DNA, making mutation for resistance very quick and fast to be passed on. There are two different methods on how this resistance is transferred from one bacteria to another. We have horizontal and then we have vertical. First off with vertical transmission. For vertical transmission, this is when during vertical transmission, the mutated gene in red in here is passed on to the daughter cells after its reproduction. And that reproduction that's happening in here is called as binary fission. Binary fission is just technically mitosis occurring in bacteria. Next one, horizontal gene transmission. So in horizontal gene transmission, we have here a bacteria with the plasmid with mutation. And in order to transfer this one to an adjacent bacteria, there would have to be conjugation happening. Conjugation means temporary union of two bacterium or two microorganism for DNA exchange. So as you could see in here, this plasmid from the bacteria is replicated. Then there is a channel or a tunnel in between the two bacterium where the new or the synthesized copy of that mutated gene will be passed on. So in the end, both of these two bacteria will now have the same mutation for the antibiotic. You can test antibiotic resistance in a bacterial culture. Bacterial culture will be done in a dish. And you have here filter papers that is submerged in the antibiotic solution. It could be several types of antibiotic or it could be different concentrations of the antibiotic. And from there, this red or is it brown area in here is where you can find the bacteria. So this is an example of the one done in the laboratory. They used E. coli in this bacterial culture. And you have here several filter papers that were submerged in different types of antibiotic. So as you could see, there are spaces in between the filter paper with antibiotic and the bacterial culture. The bigger the diameter of this point to this point, it means it could kill more bacteria. But for example, like this, bacitracin, there is no clear area around it, right? It means that this antibiotic does not have any effect on the E. coli. What should we do to reduce the occurrence of resistance? One, using antibiotics only when appropriate and necessary, and not prescribing them for viral infections. Number two, reducing the number of countries in which antibiotics are sold without a doctor's prescription. Meaning you cannot just ask it directly from the pharmacist. They would ask you for the prescription note from the doctor. Avoid the use of so-called wide-spectrum antibiotics and using instead an antibiotic specific to the infection known as narrow spectrum. So of course you have narrow spectrum means is more targeted to only small number or of species of bacteria. Number four, making sure that patients complete their course of medication, even if they say that they feel okay already.
They ask you how you are, and you just have to say that you're fine when you're not really fine, but you just can't get into it because they would never understand.
And there are still like many days left of their medicine intake, they still have to take that one. For example, let's say I have tuberculosis and then I suddenly feel that I feel good already. I don't think I have to take the medicine anymore because that will just include or will just create bigger expenses, right? That is a wrong mindset. If the doctors say that you have to take it for 7 days, you would take it for 7 days, not just until you feel better. You can only change or remove the antibiotic if the doctor says that you are allowed to do so. Next, making sure that patients do not keep unused antibiotics for self-medication in the future. People, self-medication is no-no. I know it's been a fad right now to diagnose yourself with Google, but that one is not a good practice. And lastly, changing the type of antibiotics prescribed for certain diseases so that the same antibiotic is not always prescribed for the same disease.